speaking in the session? Okay. That.
I can probably.
I just want to double check I can absolutely pronounce your names.
Yeah, so it's Saab.
Again. And the Nino?
Yeah, well, I mean, I say, I took a month off. I started taking 3 months off until 2 months off.
Hi, I am Pat.
Lovely to meet you. I'm Phil. Capital
Yes.
I went over for a week's holiday instead of 5 weeks. Just look at annual mail.
Does anyone have any favourite questions they'd like me to ask?
And then we went to
I know people here, so we can choose, it's fine.
Nobody
says it.
Psychology PhD, and she's taking that.
Well, particularly if you want me to avoid asking questions about methods, I will. But if there's any, like, if you'd rather like have a broader ecological question, or it's unlikely that I will need to ask because it's. I've noticed that the ES has been very good this year for questions. If you want an easier time for anything, just please say.
Phil, do you have a roaming mic that you need questions doing for, or are they just going to...
Yeah, I feel that.
You don't need to be recorded or not?
I just honestly don't know if this is being recorded. I haven't been told anything. Honestly, I think just pointing and shouting, it'll be fine. Roaming mic feels a bit too much like a television talk panel show.
I think it'll probably work out.
I imagine it works quite well for plenaries, but
Yeah.
this is a small enough room. People can bellow.
Okay, so 2 papers. That was my intention.
I'll allow on phone. Do you have time?
Yeah, it's quite exciting. When did you put that on? Have you put that on? I don't know, because I don't know how you or about the place. Nobody else. If you're online, which is where I found this out. That's wrong. We need more methods. Sorry, you please. Take out my phone. Yeah. Super. And James was sort of saying that actually a lot. Just sort of find that big there, so we do so we get kept up with or whatever. And then he just seemed to confirm that. He said that he mentioned that to me, you know, the jobs after.
Too short.
He should have been.
It's really short.
Then there's other people that are actually do such a waste.
So I'm assuming Kate's downstairs. Has anyone seen Kate Randall this morning? Kate, sorry. Has anyone seen Kate this morning? Not yet, but we have 8 minutes. It's fine. I think between us we all know enough about microbial black boxes to be able to. Morning. 12 minutes for talk, 3 minutes for questions. I'll use self-hotel. Good point. Did yesterday. No point. Give me. I'll have a look around. Unfortunately, no tracks today. Mouse pointer would be fine. Wild gesticulation is my favourite.
I was suggesting that's
A pointer would be cracking if it's not available. But can't find them. Yes, well, so, as the end cracking on the same side as Marsh yesterday. It's a good one. It's relaxing. Might be having a point. A pointer might be alright. So I'll be giving the traditional 5 minutes, 1 minute, and then I'll be very polite and British about gently. And sorry? Well, so if you're in a real flow, then it's really rude to interrupt you. Yeah. Like if you go a bit over and each have fewer questions. I see a clicker. Sorry guys, no clicking. It's just tapping or pressing page down. It's alright. I don't think I've ever seen one that isn't broken. I reckon that's good to go. Good morning. morning, everyone. Welcome to the BES's Microbial Ecology session at Liverpool 2016. I'm particularly happy to be chairing this session today. My name is Donkersley. I'm a microbial researcher like most of you, and I'm very happy to see the BES recognizing the importance of microbial ecology with this new session. So I'd first like to welcome up onto the stage our first speaker, Kate Randall, who's going to talk to us about using please come on, microbial communities to monitor the effects of climate change in the Arctic. That's the right one, isn't it?
Okay, good morning everyone. Thank you for giving me the opportunity to speak to you today. My name's Kate and I'm from the University of Essex, and I'm going to be talking to you about some work we've been doing of incorporating the microbial black box into climate warming research. Now we all know that global surface temperatures are rising, whether it's the most extreme scenario or the less extreme scenario produced by the IPCC report, and we need to know how these are going to affect our ecosystems. Now, we're all in the microbial ecology session, so we all know how important microbes are as being key drivers of biogeochemical cycles, those that underpin larger ecosystem processes. But when we look at the current ecological studies looking at ecological responses to warming, the microbial component is currently underrepresented. But if we're going to get a handle on how ecosystems are going to respond to warming, we need to adopt a more comprehensive approach, and that has to include microbes. And we're now at the point where we have the tools available for us through the advent of molecular techniques and high throughput technologies to be able to characterize and quantify microbial communities within environmental samples with relative ease. And actually, I'm part of a much larger multidisciplinary project entitled Impacts of Global Warming and Sentinel Systems from Genes to Ecosystems that is tasked with trying to achieve this. It's a big project that spans spatial scales as well. And it's headed up by Professor Guy Woodward at Imperial College London. And we focus on freshwater ecosystems. And in terms of the microbial communities, we really don't know, there's quite a limited amount of information on the biodiversity of microbes within these systems. But what we do know is that as a whole, the biodiversity of these systems in response to warming and global warming is declining at a faster rate than the marine and terrestrial counterparts. And although they may form just a small component of the Earth's water systems, they're a significant habitat and a valuable resource. So it's really important that we understand the potential repercussions of global warming on these systems. So today I'm going to be talking to you about microbial responses to warming at the large spatial scales. And in-situ experiments are desirable because they give you a more representative overview of what's going on. But currently, in terms of the microbial responses to warming at these levels, we have quite significant knowledge gaps. And this is because of the fundamental complexity of natural systems. There's a number of compounding factors. You have your environmental and ecological factors and interactions with those. And actually the effect of latitude as well that can confound the studies. So this needed to be tackled experimentally, and I'm going to present to you, hopefully, a way that we're going about trying to achieve this. So we really want to ask the questions, are there deterministic microbial shifts along a natural warming gradient at a global scale? And are there similar shifts across geographically separate but environmentally similar regions? And so the way we're doing this is to take advantage of natural laboratories in the environment. So around the Arctic Circle, there's a hotspot of geothermal activity. And this has generated a gradient of streams that have been exposed to geothermal activity at a range of temperatures. And 5 sites have been identified around this region. So we have Alaska, Kamchatka, Svalbard, Greenland, and Iceland. And they form a good model system because we know the Arctic is warming at unprecedented rates, so it may inform us on how other freshwater systems will respond to warming in the future. And within these systems, and this is really the key thing, is that temperature has been found in previous research to be the main environmental gradient that's happening here. So there's no effect of those compounding factors, and they're all at the same latitude, so that also isn't contributing to the data. And so it gives us the opportunity to be able to look at the effects of temperature in the environment and also to ask questions about geographical regions as well. So in 2013, a preliminary study was conducted by some of my colleagues that were looking at the microbial phylogenetic responses to warming across these Arctic geothermal gradients. So the 5 sites that I mentioned previously were sampled, and from each site there was about 6 to 11 streams, and the sediment was sampled from these streams at 4 replicate points within the streams, and there was a temperature gradient ranging from about 2 to 40 degrees. So some of my colleagues then extracted the DNA from these sediment samples and prepared libraries for next generation sequencing to profile your archaea, your bacteria, and your eukaryotes. And then the data came back, and then taxonomy was assigned to these amplicon libraries. And I'm going to present to you some of that data today. So to the data, general linear mixed effect models were applied in order to look at the OTU richness responses across Arctic geothermal gradients. So this graph here shows you the 5 sites at the top, and along the side here you can see your archaea, your bacteria, and your eukaryotes, and OTU richness along the side over there. And what I wanted to show you was that we see within the 3 sites, the Alaska, Greenland, and the Kamchatka, your archaea, your OTU richness responds positively to temperature. In the same sites, your bacteria form a unimodal response with a peak in OTU richness around 10 to 20 degrees. Whereas across all 5 sites, your eukaryotes show a decrease as in OTU richness with temperature. What I also wanted to point out was that there was 2 sites that appeared to be doing something slightly different, and that was Iceland and Svalbard. These are more isolated of the 5 sites, where your archaea seemed to show a different response to temperature with a decrease in the OTU richness. And then from them, we applied temperature abundance models to individual OTUs in order to try and see the thermal response of each individual OTU to temperature. And then we put all those together for each individual group across the 5 sites. And we found that archaea have increased thermal optima in comparison to bacteria and in comparison to your eukaryotes. And if we think about what we know about the thermal performance of these guys, this kind of made sense, so it's quite interesting. And then, so we're seeing these differences in these taxonomic groups. We wanted to think, well, maybe there's some functional implications associated with that. So this is really where I come in. And so in summer just gone, we began our more in-depth sampling of the original 5 sites. We went back, and this summer we've been to Alaska, Greenland, and Iceland. And really, to answer these questions, I was looking to target key functional genes that we know that microbes are associated with, and look at this at the population level, but also that that's been actively expressed within the RNA. So this time it was a slightly more in-depth sampling strategy. We have 12 to 15 streams per site, and the temperatures range from 3 to 36 degrees. And there's actually now 10 replicate samples per stream. So I sampled the top 2 centimeters from the sediment using a spoon and homogenized the sample, placed that into tubes, and then also had a separate tube that had an RNA preservative in it. So colleagues of ours had found in 2011 that in mesocosms that were warmed and remained ambient, there was a significant increase in the methane production from mesocosms that had been warmed. So this graph here just shows temperature is increasing in the direction of the arrow, and your methane production is increasing. So I wanted to see whether this was, how this related to the microbes that I was going to find in my field level samples. So if we go back to what we know about carbon cycling and the microbes involved in that, in particular methanogenesis and methanotrophy. So the archaea, which were found in our field samples, have a higher thermal optima. They are involved in the methanogenesis, so the production of methane. And there are bacteria, mostly bacteria, that are involved in the methanotrophy of methane cycling. So we also know that there are methanotrophic archaea, but I wanted to pull apart the functions of these, and there are key functional genes that you can target involved in methane cycling for the microbes. So you have your MCRA gene for your methanogens and your PMOA gene for your methanotrophs. So I've been in the lab now and extracting the DNA and doing some qPCR on these genes. So we want to see if there's differences in the abundance of methanogens and methanotrophs in response to warming. So that's what I've been doing recently, but I've only managed to do this since we got back from the Icelandic sediment samples. And so this graph here just shows you on the y-axis, you've got mean gene copy number of the MCRA gene and your PMO gene per gram of dry weight sediment. Along the bottom, you have your mean stream water temperature. And the yellow dots at the bottom are your MCRA gene copy abundance. So that's for your methanogens, and the blue squares are for your methanotrophs. So in these Icelandic sediment samples, we don't see a response to temperature in our methanogens as we would have perhaps predicted based on that what we're seeing in the mesocosm experiment. So just to summarize briefly, is that we have shown that we've seen differential responses across the Arctic geothermal gradients across the 5 sites in terms of the OTU richness of the archaea, the bacteria, and the eukaryotes. And we've also seen differences in the thermal optima of these guys. We've also shown that there's a potential geographic influence for your Iceland and Svalbard sites. And if we think about this, so your Icelandic site, you're seeing a reduction in the OTU richness for your archaea, and because we know that these guys are involved in your methanogenesis, maybe that may imply why my qPCR data for the methanogens doesn't respond to temperature the way I thought it would. So my future work now is to go back to the lab and to quantify the PMOA gene and the MCRA genes, your methanogens and your methanotrophs across all of the rest of the remaining sites, and to target this looking at the population, so your DNA, but also looking at the RNA, so what actual expression of genes is occurring across these sites. So thank you very much for your time and for listening, and for my friends, work colleagues. Thank you very much.
Thank you very much. Does anyone have any questions for Kate?
Light plenary speaker, also first thank you for the nice talk. And in the light of the plenary talk this morning of Manukau, could you comment on the usage of OTU richness as a metric of diversity?
I didn't actually go to the talk this morning. But I know there's other ways to do this as well. So, but we can. I just was presenting some of the data. We can look at other ways to do that as well. I don't. Yeah.
Okay, no, thanks.
Sorry.
Anyone else? Question? I'd actually like to follow up on that.
Yeah.
I was wondering if what level of identification, like in terms of phyla, family, order, what you've managed to achieve with your sequencing technology.
We went down to the genus level. Yeah, went down to the genus level.
I really like that you're using the national laboratories to study this effect. I was wondering if you thought about how it compares to streams that have natural temperatures in the range between 5 and 40 degrees that you are using in the Arctic regions. So do you have an idea how the functions that you've included in this study would work out if you take streams in Europe or in more tropical regions?
We don't have any natural laboratories like that we can sample, but we do have mesocosms around in other temperate zones that have set up that are being exposed to warming. So we could use that, but not along that same gradient of temperature, no.
Can I give another round of applause for Kate, please? Next up, we have Dave Clark. Awesome. He's going to be talking to us about the functional and taxonomic diversity of fungal organisms in salt marsh.
I'd like to start off by saying me and Kate didn't coordinate our outfits in any way this morning. So thank you all for coming to my talk. My name's Dave Clark, and I'm a PhD student at the University of Essex. I'm going to talk to you today about some work myself and some colleagues did looking at the ecology of fungi in salt marshes. So if you've looked at the programme for tomorrow's talks, you may have noticed a talk that says biodiversity is not related to ecosystem functionality, which makes my first slide awkward. For the purpose of getting through this talk, I'm going to say yes, it is. So generally, it's fairly logical that if you add species to an ecosystem, eventually one of them will bring along some new functional capability, and thus your ecosystem will have gained functional capability. And conversely, if you take species away, eventually you're going to lose some functional capability from your ecosystem. And there's been loads of really good work looking at this, particularly in plant communities by the likes of David Tillman and colleagues. And this has been, like I said, it's been really well shown in higher organisms. And that then puts emphasis on the question of what things drive and maintain diversity in communities. And again, this has been really well done for higher organisms, but we don't know a huge amount about what drives microbial diversity and community structure, and in particular, fungi. So why worry about fungi? Well, fungi are taxonomically and functionally incredibly diverse. Fungi in the environment can be parasites, pathogens, predators, beneficial endosymbionts, degraders of biological materials. So they have huge effects on whole ecosystems, and particularly so these guys, the AM fungi, which associate with plant roots and can have really big, significant effects on plant communities. So what do we know about fungal ecology? Well, we know that they have biotic and abiotic drivers of diversity. My supervisor would kill me if I didn't get one of his papers in here, so I'll mention it. But we know, for example, that pH can be quite a big driver of AM fungal diversity. And we know, thanks to other studies by people who aren't my supervisor, that they also have other things like climate factors driving their diversity, as well as things like plant richness and plant identity. However, we don't really know what the relative contribution of these sort of two groups of environmental factors is. And we also have certain sort of knowledge gaps, if you like. So most of our understanding comes from kind of single site studies or global studies, and we sort of lack this multiple site approach where we look at multiple comparable habitats. And we need to really understand how generalizable these patterns of diversity trends are across sites. We also have something of a habitat bias towards things like grasslands and woodlands. Salt marshes, for example, we know virtually nothing about the fungal communities that inhabit those. And we don't really know whether different functional groups of fungi respond differently. So people often take the fungal community as a whole, and we don't really tend to go into much detail than that and see if different functional groups are responding differently. So all of these things are basically what I'm calling contexts. We don't know how context-dependent drives of fungal diversity are. So why did I mention salt marsh? Well, salt marsh is an incredibly valuable ecosystem. We have about 45,500 hectares in the UK, and about 6% of this is in Essex. Salt marsh is incredibly important as coastal protection, which everyone really likes because it's a really easy thing to put a financial value on. It's also a really good carbon sink. Apparently a healthy salt marsh is as good as tropical rainforest for sequestering carbon. And of course, what we're all interested in, they're also harbours of really high biodiversity. It's also a very vulnerable habitat, so we know it's under threat from various aspects of climate change, including rising sea levels, changing land use, and coastal squeeze. And that's why our university is part of a large multi-institutional study known as the CBES project, which has an incredibly catchy title, which I'm not going to try and talk about. But, so this is a NERC large grant, and involves studying coastal wetlands in Essex and Lancashire. And so we're kind of piggybacking this study onto that and using their study sites for this. And we also know that plants in these habitats play a functionally important role in sort of holding this sediment together, as well as supporting this high biodiversity. So from what we don't know about fungi, I think 3 fairly simple questions emerge. So what drives fungal diversity in the salt marsh? Is it biotic or abiotic factors? Similarly, what is the balance between the biotic and abiotic environment for determining the abundance, the population structure of these fungi? And finally, do different functional groups of fungi respond differently to the environment? Or do we see consistent responses of fungi within the same functional group? So I set out to answer these questions, which using, as I said, these 6 salt marshes throughout the UK. So we've got 2 locations, Essex and Lancashire. So the Lancashire sites are situated around Morecambe Bay. So we've got Cartmel Sands, West Plain and Wharton Sands. And then in Essex, we've got Abbots Hall, Fingering Howick and Tillingham Marsh. And we sampled these in 2 seasons, so winter and late summer of 2013. In each site, we took 22 sediment cores, and we extracted the roots from these, cleaned them, and homogenized them. So important to note, we're looking at the sort of root-associated fungi in these communities. We then amplified part of the ITS gene using sort of fungal-specific primers and sequenced this on an Illumina HiSeq, and that generated about 100 million reads before quality filtering. So then we quality controlled the reads, clustered them into OTUs, and then importantly we assigned function using this sort of recent piece of software called Fungild. And we use these broad kind of functional groups known as trophic modes, which include things like symbiotrophs, saprotrophs, and pathotrophs, and then fungi that fall into one or more of those groups as well. So I apologize if this looks very complicated. I assure you it's not. So for all of these models, we basically divided our variables up into abiotic and biotic drivers. And we used negative binomial GLMs to look at fungal diversity. We used multivariate GLMs to look at OTU abundances. And then we use these finite mixture models to look at functional responses. So essentially what these do is they'll regress each OTU's abundance against your environmental variables, and then they'll cluster all of your OTUs into what we call archetypes, or sort of environmental response groups, if you like. And then we use a chi-square test to see whether those environmental response groups are associated with the functional groups. So in most sites, we actually see that abiotic factors do a much better job of predicting OTU richness than do biotic factors, apart from in this one site, Cartmel Sands. The other thing to note here is that actually the ability of the abiotic models to describe OTU richness is highly variable between sites. So just because the abiotic variables are better, it doesn't mean they're doing an equally good job across sites. To answer our second question, we've got each of these dots represents one OTU, and we've got the AIC of the biotic model and the AIC of the abiotic model. So if it's below this line, it means that the abiotic model is better. If it's this side of the line, the biotic model is better. So actually what you can see is that most OTUs kind of fall along this line, which suggests that the models are doing a roughly similar job of explaining their abundance. But there's also a bit of a skew towards this sort of lower triangle, which means that for those OTUs that are really better explained by one model, it tends to be the abiotic model that does a better job. And finally, and perhaps most interestingly, we actually found a really significant association between these environmental response groups and our functional groups. So the thing to note here is that the circle size tells you how many OTUs were found to be in each of the environmental responses, on the y-axis. And the colour tells you how many we were expecting to see based on the null hypothesis of no association. So if there was no association, we expect to see all of these circles being kind of white, which we don't see. And this was true for both biotic and abiotic variables. So overall, what did we find? Well, we saw that generally abiotic variables appear to be better at describing OTU richness and abundance. However, the ability of these models to describe these two things varies significantly between sites. We also saw that we think we found that functional groups of fungi show similar responses to the environmental factors as well. In terms of how this extends our knowledge of this system, we're shown that actually when we start to think about incorporating microbes into our sort of ecosystem processes models and things like that, we really need to take a site-specific approach because we've shown that OTUs don't respond similarly in different sites, so we can't generalize that well. We've also shown that fungi appear to be functionally similar fungi respond similarly to the environment. And that's really important because in microbial communities, we tend to assume that we've got high functional redundancy and that this means that we've also got functional resilience. If all of your OTUs that are doing one job are responding similarly to the environment, if you change that one thing about the environment they're responding to, then potentially you could upregulate or downregulate an entire functional group, which is really important to know. So with that, I'll thank my co-authors, the other institutions and CBES who've contributed data and helped with fieldwork, our funding and lab groups, and thank you all for listening.
Questions for Dave?
How did you reduce the number of OTUs? Did you have any selection?
Yes. So for obviously for OTU richness, we did nothing. For the OTU abundance modelling, I think we took only OTUs that occurred in 4 or more samples out of the sort of 220 or so that we analysed. The reason being that fungi that only occur in 1 or 2 samples are unlikely to tell us much about what they're really responding to. So that was kind of a small occurrence-based filter, I suppose.
Rob?
Really interesting stuff. I want to know a bit more about how you might link the abundance of these functional groups to actual measurable functions in the salt marsh with regards to like carbon storage, turnover, things like that.
Sure, yeah. This work was done as part of one of my co-authors' PhDs, and I really only came onto it to help with the bioinformatics and data analysis and things like that. So I can put you in touch with him, who he's doing some more of the functional measurements and things like that for his PhD. So sorry, it's a bit of a wet answer, but that's more his side of the work. So I can definitely put you in touch if you're interested.
Time for one more.
I wondered whether you could say which of the abiotic factors was more or less influential.
Oh yeah, okay. So we actually found that for a lot of the things, season was one of the biggest things that appeared to be significant. So we found a lot of fungi were very differentially abundant between the 2 seasons that we sampled. For the multivariate OTU abundances, it's a bit more of a tricky question to answer because obviously you've got so many OTUs responding to slightly different things. So that's a bit of a tricky one to answer, but generally we found that winter was one of the real important things in relation to the summer abundances.
Thank you very much, Dave.
Thank you.
Next up we have Angela.
Angela, yeah.
Angela Creevy. She'll be talking to us about the diversity of amoebae associated with the forest to bog transition. Well, you can wait just a minute or so before you start.
everybody. I'm here to talk about a project that was co-supervised by Dr. Roxanne Anderson and Dr. Richard Payne. We started work on this in 2013, investigating changes in testate amoeba communities associated with peatland forest bog restoration. So this is an aerial photograph of the flow country blanket bog type peatlands, which is situated in the far north of Scotland. And in 1970s and 1980s, this area was heavily planted with our stands of non-native conifer plantations, which you can see here. So this is usually an open treeless landscape, and the forestry was planted. And in recognition of these blanket bog and peatlands in general, in terms of carbon storage and biodiversity, there are currently large-scale restoration projects to restore these landscapes back to their open treeless form. And this has created the need to monitor how this landscape is responding to these large-scale landscape changes. Just some historical background to the history of the planting of the plantations in the flow country. This is an image taken around 1968, and Billy Mackay's father recalls the Forestry Commission, the newspaper story about the Forestry Commission abandoning bog tractors all over the flow country, kind of suggests that this may not have been the right landscape to plant trees in the first instance. This photograph shows the bog tractor being recovered. It actually took 3 tractors, 28 days to recover the bog tractor, and it was said to have cost more than the tractor itself. So just some background history. So what did we do? So we sampled across a range of sites along a west-east gradient across the flow country peatlands, and we selected 4 paired open and afforested sites. And we compared this with one restored site, which was the oldest restored site, Talaheel. Okay, so the field sampling methodology. So what we've done is we sampled test surface samples of vegetation or peat from areas of relatively undisturbed open bog. And we also sampled in restored sites. This restored site was restored in 1998. And we also collected samples from the afforested areas. And the sampling design was arranged to encompass the natural variability in this sloping blanket peatland. We collected 108 samples in total. And an important aspect of the restoration of the flow country peatlands is the removal of forestry plantations to re-wet the site and encourage the establishment and recolonisation of sphagnum mosses, which are really important peat builders, really important moss genus. What we found was topography was particularly important in order to encompass the natural variability. So this is a photograph of our control site. So this is the sphagnum-rich, undisturbed open bog, and you can see the different topographies. So we sampled from hummocks, from the lawns and the pools in the open bog. Comparable microforms in the forested sites where, I wanted to have a pointer to show you, but with a plough throw. So these are these plough ridges on this right-hand side. You've got the original bog surface and then the furrows. So this is the topography that is created from the afforestation. The restored site, despite being removed of forestry in 1998, actually still resembled the forested site in terms of the topography. So again, we compared the microforms to the plough ridges, original bog surface, and the furrows. So we considered these 3 microforms were comparable between these 3 management treatments. And testate amoeba have been the focus of my research for a number of years now, and they were particularly useful model organisms for this study because it is widely recognised through paleoecological studies and more contemporary studies that testate amoeba do respond to surface moisture conditions with different morphospecies preferring more dry habitats compared with more wet indicated species. And what's important as well about testate amoeba is that they are dominant microbial consumers in peatlands, so it is suggested that they could be used to infer changes at lower trophic levels. So we used a traditional taxonomy based on morphology and used light microscopy to quantify testate amoeba communities in forested, restored and near natural open areas. So we had 3 hypotheses. So firstly, we hypothesised that there were distinct testate amoeba communities associated with a forested, restored and open bog. Secondly, that peatland restoration has shifted testate amoeba community composition back towards that of undisturbed open bog habitat, and this is 16 years post-restoration. And finally, probably most importantly from this talk today, is that we hypothesise that testate amoeba are useful bioindicators in peatlands undergoing forest to bog restoration. And we considered 2 functional traits in this analysis, and these are aperture size, which is an indicator of the trophic position of testate amoeba, and also the mixotrophy ratio. Different testate amoeba species have different modes of nutrition and energy, so some are heterotrophic and some are mixotrophic, using a combination of heterotrophy and mixotrophy. So what did we find? So this is the results of multivariate analysis. So this is an NDMS plot of showing the testate amoeba communities. So in the red, you've got the communities in the forested sites. The blue is the restored site, and the green is the open bog. And within this, we've got different symbols illustrating the different microtopographical features. So although we did find quite a lot of variability between sampling points, that was useful because it did demonstrate that in such studies like this, that it's important to sample across a range of, across this elevation gradient. However, we could accept the first hypotheses because there was a clear distinction in the communities, in the afforested, restored and open bog. And this was further validated by the results of Permanova. We found a statistically significant difference between these 3 treatments. However, for the second hypothesis, we had to reject because testate amoeba communities in the restored areas still more closely resembled the afforested areas than that of the relatively undisturbed open bog. However, what is important to note is that the drier microforms in the restored areas, the wetter restored areas, i.e. the furrows, we're beginning to resemble some of the drier microforms in the open bog habitat. So restoration has had an effect, but it's still not yet reached the control conditions. The next set of results are the results of some functional trait analysis. So on the left, we've looked at the mixotrophy ratio, and this is between the forested, restored and opened, and there was a striking difference in the proportion of mixotrophs. So this is the proportion of mixotrophic taxa over heterotrophic taxa. So we found a striking significant difference in the relatively undisturbed open bog. So what this suggests is that afforestation has altered the trophic interactions in the afforested and restored sites, which are not showing much difference at all. Next, we looked at aperture size. Again, found no significant differences between the afforested and restored sites. However, there was a significant difference in the undisturbed open bog. So what this suggests is that afforestation and restoration has reduced the trophic level in testate amoeba, and we can suggest this because in the afforested sites and the restored sites, testate amoeba had smaller aperture sizes, which could be an indication that they are consuming smaller prey. So what could we conclude were the main conclusions from this study? So we could say that afforestation has reduced the trophic level of testate amoeba communities, and this was demonstrated in the functional trait analysis. That testate amoeba communities do seem to be recovering post-restoration, but only in the wettest areas where sphagnum had recolonised. And finally, the study highlights that testate amoebae represent functionally significant bioindicators in peatlands undergoing forest to bog restoration, so they have potential use for research in this area. And finally, I'd like to thank the funders and landowners for access to site. Thank you for listening.
Does anyone have any questions for Angela? Rockaway?
Really interesting stuff.
Thank you.
This shift towards more heterotrophic amoeba, do you think this is in any way a reflection of differences in the plant community composition and how carbon is being transferred through, and maybe the autotrophic component is just not necessary when you have a different plant community?
Yeah, it's a good question, it is true. What we found is that the mixotrophic taxa, obviously in the open undisturbed bog, they require light to survive and to thrive, whereas in the so there's different abiotic factors that influence, and we did carry out a redundancy analysis, and it did show that a strong acidity to moisture gradient where open bog communities where the mixotrophic taxa were more controlled by the moisture gradient, whereas the afforested taxa were more influenced by pH and bulk density. So yeah, I think hopefully I've answered that.
Yeah, I was thinking a little bit more about like how carbon is entering the system.
Yeah.
And if you have a higher productive system from
yeah.
plants delivering a lot more root exudates, for example, things like this, then that's selected more for heterotrophic, especially with the smaller aperture size feeding on bacteria, for example.
Yeah.
Whereas in the open system, there's a lot more carbon limitation.
Yeah.
But
Yeah.
very, very dark under the these plantation forests.
I was thinking a bit about the sort of wetted area, because like
slow to come back.
Yeah, interesting.
Thank you.
Do you think that in the restored bog they're being primarily constrained by an unfavourable environment, or whether they're dispersal limited?
Well, there's kind of interesting studies on the dispersal of testate amoeba. I think certainly the larger testate amoeba species may be influenced by dispersal, but many of the smaller ones perhaps not. They could be dispersed by winds and on other animals dispersed in that kind of way. I think the key was that the sphagnum, the vegetation change. It was demonstrated in the wetter areas where sphagnum had recolonised. So I think vegetation played a major role. And another thing to mention in the restored site is there's new techniques being developed for restoration, but at the time, this site, in the furrows were actually blocked with the trees themselves. There's different techniques happening now, which will present new and unique opportunities to look at. Maybe the legacy of the management itself may have affected it. So, thank you.
Any further questions? I would quickly like to ask whether the restoration of the sites is primarily focusing on restoration of the plant life or the topography of the site, and what techniques there are for altering topography to remove these furrows and restore it to what it used to be like.
Well, the restored site Talaheel is just, it's more homogeneous, the same sort of treatment. Sorry, what was it?
What physical techniques there are for going into these sites and restoring, making them physically, topographically more like the original.
Yeah, I mean, there's new trials. It does seem to be that again. The landowners for the restored site are the RSPB, and they've conducted different trials and vegetation surveys. But the main aim is to encourage the recolonisation of sphagnum mosses, and I think I'm not really sure about the restoration techniques in terms of the topography. It's very, very large scale, so yeah, I think watch this space for that. So, thank you.
And hopefully not to lose any more tractors.
Yeah, hopefully.
Thank you very much. Next up we have Antonino, who's going to talk to us about the interactions between soil microbiodiversity and an aphid endosymbiont.
Okay, thank you very much. I'm really pleased to be here to talk to you about the last experiment of my thesis. So it is widely known that above ground and below ground communities interact and influence each other at different spatial scales and temporal scales, leading to different ecological effects, even on plant growth. So what we wanted to do is to analyze the microbial ecology of this system. So we asked ourselves two questions. One is, can aphids or aphids endosymbionts impact on soil microbial community? And the other, can soil microbial community impact on aphids microbiota? To answer these questions, we set up an experiment including 2 plant species belonging to the Solanum genera, 4 aphids clonal lines, and 3 soil communities. One, the first one directly from the field, then just the extracted mycorrhizal fungi spores, and just the bacterial community. And from this system, we took samples of aphids, leaves, roots, and soil. Just for who it's not really into metabarcoding, I will explain you very quickly what I've done. So metabarcoding is like going to the mall. We have our samples, but really we are not interested in everything it's in the mall. So, for example, food. So we extract our DNA. That it's what we are interested in. But we will not buy the entire stock of food in the supermarket. So with a shopping list that is PCR to select what we want to buy: bacteria, fungi, mycorrhizal fungi. Then with the sequencing machine, we scan our barcodes, and through bioinformatic analysis, we have our receipts that shed the light on our ideas. So what we've done is to analyze 3 communities: the bacterial, the fungal, and the mycorrhizal fungal community, through 16S, ITS2, and 18S on our different samples. So going back to the questions, can aphids or aphids endosymbiont impact on soil microbial community? The answer is yes. What it's here is just the microbial community of soil of potato plants, the domesticated species. So what we have is the presence or absence of aphids. And we see that in each of these 2 clusters, there are 2 subclusters, one with the presence of aphids and the other one without. So we can say that aphids can influence the soil microbial community. You can see 2 different clusters because this one is the big effect in this PCA is the soil microbial community. So this is one microbial community, and these are the other 2. Also, what we highlighted is that different aphid lines impact in a different way on soil microbial community. This is the entire community, the entire soil community of both plants. And what we can see in the lower part, there is only one aphid line. Why it's different is because this line hosts Helminthionella defensa. That is a protective endosymbiont that protects aphids from the development of eggs of parasitoids. So the second question was, can soil microbial community impact on aphid microbiome? The answer is still yes. What we have here is the aphid microbiome. We have 2 big clusters. This one is the aphid line hosting Helminthionella defensa. But within each cluster, we have a clear differentiation by soil microbial community. So we can say that soil microbial community influences aphids microsymbiont, in particular if in the soil microbial community there are mycorrhizal fungi. So Soil microbial community is influenced by aphid presence. We have top-down effects. Aphids bacterial community is influenced by soil microbial community. So we have bottom-up effects. And one aphid line, hosting Mediterranean defensa, influences plants microbiota in a different way. And we asked two further questions. Does plant species matter? And can below-ground microbial community shape the plant microbiota? The answer is still yes. So the plant, we have here the whole microbial community of the entire system, and is clearly differentiated by plant species. And also, if we analyze the below-ground community, we can see that it's clearly differentiated by soil inoculum. So, summarize everything, we have that plant species and aphid presence impact on leaves community. There is an error, but plant species can impact, soil and aphid presence can impact on roots microbial community, and soil can impact on, soil inoculum can impact, of course, on soil microbial community, but plant species can impact on fungal community and aphid presence on the bacterial community. The future work will be on the identification of microbial actors of these interactions, the effectors that leads to these effects, to extend the analysis to other plant species to see if this effect is conserved, to disclose the role of Mediterranean defensa that was quite strange and unexpected, and to build networks. What we want to do is to analyze the co-occurrence of each taxa in our system, and to build the networks with and without aphids to see how the different microbial taxa interact and influence each other. The last, I want to say thank you to all these great people who paid everything, and you for your attention.
Thank you very much. Does Aaron have any questions on Nino? We have a lot of time, so.
Yeah, thanks. That was really nice talk. I really liked your method coding.
Thank you.
Have you any plans to look at the nutritional status of the plants and of the aphids?
Yeah, this is what we think, because this effect on aphids was quite unexpected. So what we think is that mycorrhizal fungi can impact on plant physiology, plant nutrition, something similar, and so through the plants can have an effect on aphids nutrition and their related endosymbionts. Yeah.
Any further questions? I would appreciate if you could give, just go over again, give more detail how you assign causality and directionality between the soil microbial community and the aphids and the plant microbial community.
So,
the actual statistical method by which you assign that causality.
The statistical method?
Yeah, just because.
Yeah, so what we have done, here see the entire community, but we analyzed the bacterial, the fungal, and the mycorrhizal fungal community separately also. And this effect is quite conserved. So what we have done is to clusterize the sequences, get the OTUs, assign a taxonomy, and then we have done basically multivariate analysis and Permanov analysis to see these effects on the different communities and on the whole microbial community, taking into account all three markers we used.
Since we have a little bit more time, there's a lot of evidence that suggests that the endosymbionts inside aphids can actually effectively hide them from predators. And I'm wondering if you consider sort of the implications of that for the microbial communities of plants, actually making them more susceptible to pests by reduced pests.
Yeah, this is quite interesting because what we highlighted is that the aphid line hosting this protective endosymbiont show different impact on plants and on soil microbial community. So maybe there is a sort of influence between the soil microbial community, the aphids, and the parasitoid. But this effect was quite unexpected on soil, so I think we will need further experiment to shed light on this phenomenon probably.
Do we have any further questions? No, well, thank you again.
Thanks.
thank you everyone. We're now going to move on to Argen, who's going to talk to us about using DNA metabarcoding methods to analyze aerial mesofauna.
Hi all. I am Arjen de Groot, and I'm working as a molecular ecologist at Wageningen Environmental Research, which is part of Wageningen University and Research in the Netherlands. And indeed, I will show some results from a recent project we have been doing together with colleagues from the Netherlands Ecology Institute for Ecology, as well as Aarhus University, which dealt with using DNA metabarcoding to study community shifts in the aerobiome, which is the community of organisms present in the air. So we know that community assembly depends on both local conditions, let's say environmental filters, as well as dispersal, which may be especially relevant for larger plants and animals, but we know by now, I think, that even in the microbial world, not everything is always everywhere. And the airborne dispersal occurs throughout the tree of life. Not only bacteria and fungi are present in the air, but also a wealth of protists, as well as obviously plant pollen and seeds, and even small mesofauna like mites and collembola might sometimes disperse through the air. And what we also know is that members of different taxonomic and functional groups that arrive in a certain community interact there locally via trophic interactions or mutualisms, host-parasite relations. And it has been shown that also timing and abundance of propagules arriving to a new location is influencing those kind of relations. So one of the questions we might ask is to what extent also linked dispersal under the same kind of conditions or the same kind of factors might influence those, to what extent that exists to study, then how that might influence interrelations. But yeah, before we can start doing that and even derive hypotheses on that, it's very important to understand which species are where and when in the air and in which combinations. So the spatial temporal variation, for instance, which for air samples is actually quite unknown until now. Some work has been done since the advent of molecular techniques for bacteria and fungi, showing effects of, for instance, seasonality, land use types, effects of local nearby point sources. But for instance, the effect of weather is quite unknown, with some artists finding effects of particular atmospheric conditions, while others didn't. And especially for the larger bioparticles, protists, mesofauna, but even for plants, much less is known, especially because much of this work has been done until now based on morphological identification, which is much more time-consuming and limits our ability to process large amounts of samples from many different places or time points. So DNA techniques using multiple markers allow us to study the entire aerobiome for a larger number of samples. So that's what we set out to do in a study in October last year, where we used the so-called Hearst spore sampler, a device that sucks in air and blows it over a drum on which adhesive tape is mounted, and this drum rotates slowly, making one round in 7 days. So afterwards you can remove this tape and you have your temporal variation in your aerobiome over a 7-day time period. So we did that for 3 weeks in a row, so 21 days in total. And we did that simultaneously at 2 sampling points on 2 rooftops on the Wageningen campus, which is, as you see on the map, is located in between the urban environment of Wageningen and the large agricultural lands to the north and west. So we then chopped this strip into 7 pieces, representing the 7 days of a week. And each of those pieces was then again cut in 2, and one half was used for morphological identifications, as well as culturing of protists. That's something I will not discuss today. I will now focus on the DNA part, where we used these strips to extract DNA, and then I did metabarcoding. I expect that most of you know this technique by now, and otherwise have had already a perfect explanation in the previous talk. So we did Illumina MySec sequencing, Nextra library preparation, and used 3 different markers. The first 400 base pairs of the RBCL gene for plants, a newly developed 18S barcode to cover all eukaryotes, and a general 16S marker for bacteria. So what did we find then? To start with the RBCL marker, we found a large diversity of plant species, 29 different families in total. A large group present were the Urticaceae, stinging nettles, but also Atlantic cedar was common, which is not that surprising because at the time of sampling this was producing vast amounts of spores into the air. And in total about 60% of the total abundance was coming from species that did pollinate at the time of sampling. But the other part also was from trees and crop species that did not produce pollen at the time. And this must have been plant fragments. And indeed, in the morphological analysis, we see all kind of leaf hairs, for instance, on the tapes. English oak is an example of that. So if you go to 16S, the bacterial diversity, a large group present there were the firmicutes, as well as actinobacteria, proteobacteria, and Bacteroidetes. And if we zoom a bit more into the groups within that, we see quite some gut-related bacteria like Clostridia, Corynebacterium, and Bacteroidetes, such as Prevotella. As well as some plant-associated groups like Sphinxobacteriales, Pseudomonas syringae, so plant pathogens as well. And if we go to the 18S marker, the eukaryotes, this was dominated by Asco and Basidiomycetes fungi, mainly, not that surprising, a lot of spore-forming saprophytes, many different mushroom species, as well as over 30 different known plant pathogens, and for various potential animal pathogens. And if we remove the fungi for a while, then we see in the right graph that there's also, at lower abundances, a large wealth of different protist phyla, for instance. One of the major groups are the oomycetes, mainly Phytophthora, as well as apicomplexans, animal parasites like Gregarina, and Opalinata, and even some arthropods, which were in fact small mudflies that we also saw on the tapes. So if you now look at the temporal variation, we see that over time there is a large variation. And in some groups that is a bit less obvious, but in some groups like the Bacteroidetes or oomycetes, you see a strong pattern that is the same in both sampling locations, which are shown here in blue and red. So we wondered what is causing this. And luckily we share our building with a climatology group, which has its own weather station on the campus. So we had weather data from the same very nearby locations. And we studied in multivariate analysis the relations with those weather conditions. And using pure mutation tests, we could show that air pressure was one of the factors that was significantly related with the community composition of all 3 markers, as well as radiation, so sunlight in 18S, and minimal temperature in RBCL, so the plants. And surprisingly, rainfall and wind speed were not really significantly related to the community composition, although of course rainfall was quite connected with sunlight, inversely related, and wind speed with air pressure. So this is on a very broad scale, the full community. If we zoom in, we see that the main effect within the 18S marker was of course produced by these large amount of fungi, Basidiomycetes and Ascomycetes showing opposite effects with air pressure. while other groups did different things. For instance, the oomycetes are significantly related with radiation, so the amount of sunlight being more present on sunny days with less rain. And we found the same for some bacterial groups like the Sphinxobacteria, which showed the same pattern, while other groups like Clostridia showed the opposite effect, being especially present under wet conditions on cloudy days. So different patterns. And we now are interested to see how this, to dive into this further. And one of the things we are doing now is this kind of correlation networks that we also saw in the previous talk, showing links between all our OTUs from all 3 different markers, and only showing the links that represent a correlation of above 0.7%, correlation of 0.7 or higher. And what we see then is some quite dense network with a lot of connections. Within the middle, all the ascomycetes and basidiomycetes occurring in this sharing presence-absence in the same kind of samples. Clostridiales, so mainly the gut bacteria form another cluster, as well as the arthropods together with apicomplexans, which might suggest that these apicomplexant endoparasites were present within these arthropods. And the similar thing for the Sphinxobacteria, which were connected with not the pollen-producing plants, but the leaf fragments, the species that we found as leaf fragments. And the oomycetes formed really a separate group. So this was really a preliminary analysis, and we will explore that much further. So some key messages. We find a large diversity of not only bacteria and fungi, but also other groups of eukaryotes, including a lot of different protists, among which pathogens and potential animal and plant pathogens, parasites. Community composition was significantly related with some weather conditions, especially air pressure and radiation. We find strong correlations between taxa, both within taxonomic groups as well as between groups from very different parts of the tree of life. And more knowledge about that kind of correlations and how that varies with different places and times will benefit our understanding also of community assembly and the interactions therein. And since we have the molecular tools for that available, I think we can learn a lot in the future years to come. And with that, I would like to thank you for listening and some other people that were involved in this study. Thanks.
Do we have any questions? It's the back.
Yeah, I just wondered if you showed some differences or apparent differences between, particularly in the plant communities, I think, between your east and west capturing points. Is that partly related to the landscape that's around the east and the west? Do you have different landscapes? Because it looked like you had sort of agricultural areas on one side and perhaps peri-urban, urban areas on the other side.
Yeah, it might be. It might be related to wind direction then, especially as well.
Did you see any of that distinction, so like distinction between crop plants and I don't know, garden plants or something, for example?
No, not really at that level between the different locations. No. But yeah, especially the relation with wind direction is something we need to explore further.
Yeah.
Is it the back of the right again?
Thank you, very interesting talk. I have a question about whether you tested or considered to test any of the properties of the properties, for example, the spore size or the shape. In other ways, like are there some traits of the spores or the properties that would make them better floating in the air or better dispersing?
Yeah, that's really one obvious thing that we really want to do, but we are still working on that, gathering the data, that kind of data for all the species.
Thank you.
Ross at the back.
It's more of a technical question. You had quite a size difference between your apricots for your MySpec sequencing. Did you sequence those together, or did you sequence them on different runs?
Those were sequenced together.
And did you notice whether or not the 18S was as covered as well as the RBCL and 16S, because it's really small?
Yes, Yeah, and we were aware of that, and it was indeed well covered. Yeah, we were happy to see that. We were a bit afraid of some problems there, but luckily it wasn't the case.
Yeah, we have time for just one more.
One question. In your protocol, I was wondering how you actually extracted DNA, because you're going from like bacteria communities to fungus spores in the same tube, and I was wondering what kind of procedure you used to extract DNA from all of those types of organisms.
Sorry, I didn't get the first part.
So what protocol did you use to extract the DNA from your entire community sample?
We used a mobile plant extraction kit, including a bead beating step. So you roll your tape into the tube and add beads, and then yeah.
What beads did you use?
Silica beads, I think it was. Optima.
Thanks.
Thank you very much. Yes, next up is Path, who's going to talk to us about the microbial communities associated with water distribution networks in broiler chicken farms and their impacts on Campylobacter.
Hello, my name is Paz. I'm a PhD student in the University of Salford. I'm going to talk to you today about my PhD work characterizing the microbial communities associated with the water distribution system of poultry farm and their role in Campylobacter infection. So I'll introduce you to Campylobacters. So Campylobacters are gram-negative bacteria. There are around 21 species in the genus, but not all of them are pathogenic. Campylobacter jejuni and in second place Campylobacter coli are the main species responsible for foodborne infections in humans. They are microphilic, thermophilic, have really complex nutritional requirements, so they are usually considered fastidious. But they show a very complex epidemiology with extensive animal and environmental reservoirs, including wildlife and farm animals, and also water. And they infect 208,000 people each year in UK, costing £900 million to the economy. It's been estimated that up to 4 in 5 cases in the European Union can be related to the consumption of contaminated poultry meat. And the Food Standards Agency has reported rates between 50 and 80% of positives at retail in the last 2 years. This just shows the amount of GP presentation cases with infectious intestinal disease, and the ones in blue are the ones caused by bacteria. And we can see how Campylobacter is really the main bacterial cause of infectious disease in the UK, which is also true globally. So the chickens are born, and they are Campylobacter free. And then they go to the farm, and they usually get Campylobacter when they are around 2 weeks old. So there's loads of possible sources that have been identified and studied, but the question remains of how and why the chickens get Campylobacter in the first place. So the research I'm going to present today tries to understand the role of the water distribution system in this complicated picture. So research suggests that Campylobacter might survive in the environment by interacting with other members of the microbial communities, in particular with microorganisms growing attached to the pipes as biofilms, and protozoa can have a protective effect against environmental stresses for Campylobacter. But we really don't know a lot about the microbial communities in the water distribution system of broiler farms. So the aim of the project was to characterize these communities and to assess the role in Campylobacter infection. So to do so, we conducted a 7-week pilot study in a conventional broiler farm in the UK. This is an schematic representation. So the part over there is where the chickens actually are. And when you see the part between the biosecurity barrier and the actual shed is what I call the anteroom. So samples for sequencing were collected from the water, both inside the chicken shed and in the anteroom, and then to sample the biofilms from the internal part of the nipple drinkers, and also by swabbing inside the pipe, and also the external part of the nipple drinker was also sampled separately. And then Samples were collected to try to isolate Campylobacter by culture, and this included feces, the nipple drinker basket that you can see in that picture over there, the boot socks, which is what I had to wear over my wellies to get inside the chicken shed, and water, both, again, from inside the chicken shed and the anteroom. And this was done weekly for 7 weeks, which covers a whole cycle. So week 1 is before the chickens were placed into the shed, and week 7 is just after they had already left for slaughter. So I just wanted to show you some key conditions that change across the cycle. So temperature decreases, and the flow rate of water increases as chickens get older and bigger. And other key points are the administration of lincospectin early on, which is a combination of lincomycin and spectinomycin, CVAC, which is a vaccine that is also distributed through the water, and the process of thinning, which is when some of the chickens get sent to a slaughter before the rest of the flock, and is known risk factor for Campylobacter colonization. And the pH remained stable and around 7 through all the 7 weeks. So really quickly, the methods for Campylobacter isolation, it was enriched in selective broth and then plated in selective agar, and then DNA was extracted to detect it by molecular methods. And to study the wider microbial community, I did 16S and 18S amplicon sequencing using primers from the Earth Microbiome Project, and I seq in situ in the University of Salford, and then sequences were processed in CHIME, and statistical analysis were carried on in Primer 7. So this shows multidimensional scaling plots of the prokaryotic and eukaryotic communities. So each stot is a different sample. The symbols correspond to the different sample types. So really quickly, the blue triangles is the water inside the chicken shed. The red triangles is the water, the source water from the anteroom. The green squares are the internal part of the nipple drinkers, so the biofilms. The red blue circles are the biofilm collected with the swab. The pink diamonds are the external part of the nipple drinker, and the crosses are just negative controls. So we can see that, generally speaking, both representations tell a similar story. So we can see how samples cluster mainly according to their sample type. But the analysis, these two analyses have a correlation of 0.59. So they do tell a similar story, but not exactly the same one. And actually, it really doesn't make a lot of sense if you are interested in the ecology of the water distribution system to have this separation. So I joined the data together and carried on with the analysis. And I wanted to test if these differences that you can see in the MDS plot, but also when doing cluster analysis between different sample types, were statistically significant. And I was also interested in knowing if there were any temporal differences across the 7 weeks that maybe are not so evident from the plot. And by a two-way cross-anasem, we can see that both differences are significant. So I have joined the trajectory across the 7 weeks in the MDS plot to try to illustrate this. And it's not so obvious, but you can see how there's some patterns that are the same across the different sample types. So there's like a big change in community between week 1 and week 2. And the samples by the end of the cycle seem to be closer together. Week 1 is always in the right, in the right of the plot, like that. And you can also see it in the cluster analysis because the samples, again, cluster according to sample type, but usually the samples that are closer together are from consecutive weeks. So anyway, it's pretty obvious that the main differences are for different sample types, so I wanted to analyze that a bit further. So I did a one-way anasem, and I wanted to have a look to the difference between particular sample types. So if you look, well, for those of you that are not very familiar with the R statistic, the higher the value, the higher the difference between the communities that are being compared. So I think it's very interesting that the source water and the water inside the chicken shed groups have a really high R, so show very different communities. I hope this works. And you can clearly see that in these 3D MDS, how they cluster different. Also, the water inside the chicken shed seems to have a different community than the biofilm samples as collected with both methods. But in contrast, these two biofilm methods, so the two groups of biofilm samples don't seem to be significantly different. And we can also see that in this plot, so we can see how the water, the pool water clusters independently of the two biofilm samples that are mixed. And going back to the pool water, this heat map shows the most abundant OTUs, and I think it illustrates both how the source water is and the water inside the chicken shed have different communities, but also how the communities actually do change across the cycle, across the 7 weeks. So we can see in the pool water how weeks 3 and 4 show like quite similar profiles, and weeks 5, 6, and 7 also show quite similar profiles. And that's mainly due, in this case, for an increase in lactobacillus and staphylococcus and ploemia towards the end of the cycle. In terms of Campylobacter detection, it could not be isolated by culture. Campylobacter-like colonies were isolated in week 6, but they were finally identified as Helicobacter pylori. So Helicobacters are closely related to Campylobacters. Helicobacter pylori is actually an emerging enteric pathogen. It has been related with digestive pathologies, including gastroenteritis. And it has been previously isolated both in chickens and in chicken meat at retail, but there's no definite proof that a human infection can come from the consumption of chicken meat. From the 16S amplicon sequencing, we identified Campylobacter sequences in 2 samples, so in week 5 and in week 7, across different analysis platforms. And interestingly, samples in week 5 were identified to genus level, to species level, as Campylobacter urolithicus, which is also emerging pathogen that has been detected not long ago in the feces of patients with gastroenteritis in the south of Ireland. And it had never been detected in association with chickens before. It had only been detected in association with cattle. And these sequencing results led me to investigate a little bit further by PCR. So it was also detected by PCR in week 6 in the water and in the feces of the chickens, and in week 7 in the water. And the qPCR for Campylobacter jejuni was negative, in agreement with the culture results. So to conclude, I think the microbial communities in the water distribution system differ from those of the source water, are niche-specific, and change across the rearing cycle. I think this illustrates how important it is to look at both prokaryotic and eukaryotic communities, but also to sample the pool water and the biofilms if we want to understand, get more information about the ecology of a water distribution system. And I think it kind of shows that the water distribution system has the potential as either a source of vehicle of Campylobacter infection of chickens. The farm that I've been sampling has really high biosecurity standards, and it only encounters Campylobacter very occasionally. I think it would be very interesting not only to repeat this, but also to repeat it in farms with a higher incidence of Campylobacter and see how the whole microbial population compare to the other farms. And that's it. Thank you for listening, and thank you to everybody in the slide.
We have time for one question if anyone's got anything. I'd like to quickly ask, you say that there's an infection of Campylobacter in the chickens. Do you notice any fitness costs to the infection, like what the actual diagnosis could be?
Yeah, Campylobacter is a conditional pathogen in chickens, so they are mostly not very bothered, but in certain conditions, they can gain less weight and be more prone to other infections. But in the farmer sample, the farmer says that he doesn't know, he cannot tell.
Thank you very much.
Thank you.
Next up we have Sarah Koenig to talk to us about the responses of microbial ecosystem function to disturbances.
Okay, yeah, thank you. Today I will talk about collapses. Understanding drivers and thresholds for collapses is certainly an important issue for many different disciplines, as we also see in this nice example, but especially, of course, for in the field of ecology. So we have an ecosystem which is delivering several ecosystem services and is influenced by biotic and abiotic factors, and often it is also exposed to disturbance events of different kinds. How good this ecosystem and its functions can handle these disturbance events depends on the stability. And if the system is not stable enough, then a collapse may occur. So the question now is, what brings the ecosystem to the edge of collapse? One thing may be that disturbance events may be too frequently, or the effects of these disturbance events may be too harsh. So disturbance characteristics are certainly an important factor which we should have in mind. And the other thing is the biotic and abiotic factors, which may influence the ecological parameters. So, for example, the resource availability, which is influencing the growth rates of the organisms. In this study, we've focused on one example of the terrestrial environment, more specifically the response of microbial ecosystems and the function of bacterial degradation of organic pollutants, responding to recurrent disturbance events. We focused here on the frequency and the spatial occurrence of the disturbance and on bacterial dispersal and bacterial growth. So the aim of this study was to identify key factors of the resistance of microbial ecosystem functions with the example of biodegradation of organic pollutants under recurrent disturbances, and to determine drivers and thresholds of functional collapses. Okay, for analyzing these different factors which may have an influence to the collapse and to the functional response, and to systematically vary different parameters of interest, we've used a bacterial simulation model, which is called Ecology. This model is population-based and describes the resource consumer dynamics of bacteria and substrate, which is the pollutant of interest, which should be degraded, basically. This model is spatial-temporally explicit, visualized on this circular area with a diameter of 88 millimeters, consisting of around 6,000 habitats. Different processes are described within this model. The substrate is able to diffuse on this whole simulation area. There's substrate input in each time step, depending on the current substrate concentration and the maximum substrate concentration, representing subsequent dissolution of absorbed pollutants in the soil system. The substrate is reduced when the bacteria are uptaking this substrate, which basically describes the biodegradation performance. The bacteria are also able to disperse within the simulation area, and the bacterial concentration is reduced if not enough substrate is available for the maintenance of the bacteria, and due to a disturbance event. So for implementing such disturbance events, we first of all generated an undisturbed reference state in which the bacteria and the substrate are homogeneously distributed on the whole simulation area, and the incoming substrate exactly matches the maintenance of the bacteria, such that the bacteria are not able to grow, but they are also not dying. Then a randomly distributed disturbance is applied on half of the simulation area. So in this black area here now, the disturbance has occurred, and the white area is completely undisturbed, with the effect that a huge amount of the bacterial cells is removed on this disturbance area. We can vary this disturbance regime on a spatial way by varying the degree of fragmentation of the disturbance, for instance, with a more clustered disturbance pattern or a higher fragmented disturbance pattern, and in a temporal way by varying the disturbance return interval. Okay, so how does this look like? Plotted here now is the biodegradation performance in relation to the undisturbed reference state. So in the beginning of the simulation, the biodegradation performance is 100% because the system is completely undisturbed. Then the first disturbance occurs in this example now with this disturbance pattern, which is a non-fragmented disturbance pattern. The function goes down to around 50% because half of the area is disturbed. Afterwards, the function has time to recover until the next disturbance occurs after 80 hours in this example. So the next disturbances always occur with another disturbance pattern, but always these disturbance patterns has the same characteristics of this degree of fragmentation. So it goes on. And at some point, as we can see here, the system can not further handle these disturbance events, and the function collapsed. Okay, so you may think now that these collapse depends somehow on the time the function has between two disturbance events to recover. This is what we tested first of all. Here I've varied the disturbance return interval length between 5 and 150 hours in steps of 5 hours and calculated the time to collapse. And as you can see, we have a clear dependence on the disturbance return interval. So at some point, over some disturbance return interval length, the function does not further collapse. This again was for these non-fragmented disturbance patterns. If we now increase the degree of fragmentation of these disturbance patterns, then we also have a dependence on the disturbance return interval, but the threshold for collapses is shifted to lower disturbance return interval lengths. So we have also independence on this degree of fragmentation of the disturbance events. Okay, what does this mean now for the biodegradation performance? In this example, now we've varied 4 degrees of fragmentation between these not fragmented disturbance patterns and highly fragmented disturbance patterns, and again, disturbance return intervals from 5 to 150 hours. Plotted is the total degraded substrate within 2,000 hours of simulation time in relation to the undisturbed reference state. And these widths of boxes are indicating the time to collapse. So if a box is only half full, then the function has collapsed after 1,000 hours of simulation time. And as we can see, the disturbance return interval length has also an influence to the biodegradation performance. So more substrate is degraded if the disturbance returns more seldom. But the degree of fragmentation has a higher influence. As we see is with these highly fragmented disturbance patterns already with 50 hours of disturbance return interval, we have biodegradation performance which is the same as with the undisturbed reference state, and we also see collapses only up to 25 hours of disturbance return interval length. Okay, so we have an influence of the disturbance characteristics to the biodegradation performance and also to the collapse probability. But what is with the ecological parameters? So we've varied the growth rate and the bacterial diffusion coefficient and simulated combinations of 3 of these parameters each. In this example now with a disturbance return interval of 50 hours and lowly fragmented patterns. Plotted again the degraded substrate within 2,000 hours of simulation time and the time to collapse indicated by the width of boxes. With increasing y-axis, the growth rate is increasing and bacterial diffusion coefficient is increasing from left to right on the x-axis. And as we can see, the ecological parameters have an influence both on the biodegradation performance and on the functional collapse. So in the best case, with high growth rate and high bacterial diffusion coefficient, we have a really high biodegradation performance and no collapse. And in this worst case, we have a really low biodegradation performance, and also we've observed a functional collapse. We also see that the influence of the growth rate is higher as the difference in this direction is higher as in this direction. Okay, we've done this for many other disturbance regimes. On the outer x-axis, these disturbance fragmentation degrees you already know, no to highly fragmented, and 5 different disturbance return interval lengths, 20, 50, 80, 150, and 250 hours. And the small plots are the same as in the slide before. In this example from the slide before, we've identified an influence of ecological parameters, as it was also for all of these disturbance regimes, which we identified to be moderate in their combined effect of the fragmentation and the disturbance return interval. Under this disturbance regime, we have observed no substantial influence of the ecological parameters because this disturbance regime was too severe, so it does not matter how good the bacteria can grow or disperse. And in all of these disturbance regimes, we've also observed no substantial influence because the effects of the disturbance was too weak. So the influence of the ecological parameters highly depend on the specific disturbance regime. Okay, to sum this up, we've seen that bacterial growth and dispersal are key factors for the functional resistance under recurrent disturbances, but highly dependent on the specific disturbance characteristics, here namely the degree of fragmentation and the return interval. Thresholds for functional collapses should be identified for specific disturbance regimes, and enhancing bacterial growth or dispersal may be an option to prevent functional collapses. Okay, so we're at the edge of collapse. It depends on how fast you can run and how good you can reproduce, but this only matters if the disturbance is not too harsh. Okay, with this I would like to thank all of these people and my colleagues of the Department of Ecological Modeling and Environmental Microbiology of the UZ in Leipzig. And before I thank you, I would also like to draw your attention to a session which I'm organizing with colleagues from the University of Stockholm and Vienna at the EGU in Vienna in April next year. Okay. Thank you.
Do you have any questions for Sarah?
So you mean adaptation of the parameters, of the ecological parameters or something? No. No, no. So, no. This would certainly a really important point, so to include adaptation in some way that they are, especially if the disturbance is varying as it is, and always randomly occurs on the same habitat. This would certainly be a really interesting point, but it's not included in the model.
Anyone else have a question?
Yeah, if you take these results back to reality, let's say, what are the kinds of, maybe I've missed it, but what are the kinds of disturbances that you're thinking of, the kind of ecological functions of bacterial communities that you're thinking of that this model would capture?
You did not miss it. So the idea behind this, when we think of the reality, is from the disturbance type that it's for instance, some toxic chemical is coming into the soil system, and we chose to vary the spatial occurrence because of simply heterogeneity in the soil system. So we have high heterogeneity, we have high dynamics in the soil system, which is also changing the spatial occurrence of the pore size network and stuff like this. So this is, this was a little bit the motivation behind this.
Thank you very much.
Thank you.
Last up we have Susanna Bird, who's going to talk to us about the wonderful world of multi-omics approaches and the power you can get from that method.
First, I'd like to say thank you to everybody for seeing it through and staying right till the end to hear the last talk before lunch. And also thank you for welcoming me into the microbial ecology world, because I'm not actually a microbial ecologist. So I'm hoping that this talk will be of some interest and relevance to the microbial ecologists in the room. So I'm going to talk about how a multi-omics approach can inform our understanding of mixed microbial consortia. And particularly in this study, we were looking at a microbial consortia that could break down lignocellulose. For those of you that don't know what is lignocellulose, it's a composite complex polymer that's found in the cell walls of plants. It's there to provide rigidity and recalcitrance. It's made up of 3 components: cellulose, hemicellulose, and lignin. Cellulose is a crystalline form of glucose with beta-1,4 linkages, and these create crystalline microfibrils. These are surrounded by hemicellulose, which is a more structurally complex polymer. It's predominantly made up of xylans, but it's got a number of different hexose and pentose sugars, decorations and substitutions, and it's decorated with a number of different kinds of side chains, so this makes it very heterogeneous structurally. And this intertwines the cellulose microfibrils that you can see there. Ooh. And then lastly, lignin is the final component. And this is the most recalcitrant of the 3. It's a polyphenolic complex substrate that is bound together using free radical chemistry, which means that its structure is very heterogeneous. There's no regularity to its structure, and it's very recalcitrant to enzymatic degradation. So while lignocellulose is a rich source of sugars, they're actually very inaccessible in the type of form that they come in the plant cell wall. So while this is an important food source for a number of different organisms in the environment, they have come up with a complex array of strategies for breaking down this complex and recalcitrant food source. Particularly microbial consortia are found in soil, in compost, in marine sediments, but also there are some animal models that are able to produce these kinds of enzymes that are able to attack this complex structure and break it down. And these enzymes have a bioindustrial application. They can potentially be used in the generation of second-generation biofuels that aims to use inedible plant material as the feedstock rather than competing for edible human food sources. So we were interested in a composting microbiome because this is a community that exists in fairly ambient conditions, so they're able to break down lignocellulose in ambient temperatures under ambient conditions. And this is a domestic compost heap that we used. It was enriched with wheat straw for 12 months. And then we brought it into the lab. And we inoculated liquid cultures that had wheat straw as the sole carbon source, and we grew it for 8 weeks and sampled on a weekly basis. And we looked at nucleic acids, so we extracted nucleic acids and proteins on a weekly basis. We also checked that there was something growing in there, so we think these are microbial biofilms that are colonizing the internal spaces of the wheat straw. And on a weekly basis, as we looked at the wheat straw, we could visually see that there was degradation happening. In the early weeks, it still looks more or less like wheat straw, and by the end of week 8, we see more or less there's this powdery residue that looks very different. When we quantified this, we found that there was approximately 60% mass loss over the 8 weeks, with most of that mass loss happening in the first 2 or 3 weeks. And when this was quantified using solid-state NMR, these spectral peaks correspond to the different elements of the lignocellulose, the cellulose, hemicellulose, and lignin, and we saw a significant decrease in the amount of cellulose that was being reported through this assay. We also found that our community was causing an increase in pH in the culture supernatant, so from a starting pH of around 6.5, and by the end, the pH was up to about 8.5 to 9. The majority of the enzymatic activity that we could assay was in the first few weeks. But interestingly, there didn't seem to be an enrichment in the lignin fraction, which we would have expected, given how much mass loss we were seeing over the 8 weeks. So to talk a little bit more detail about the multi-omics approach that we took. So as I mentioned before, we sampled on a weekly basis and we extracted nucleic acids and proteins. So we were interested in the total meta-exoproteome. So this is the proteomic fraction produced by the culture and excreted into external environment, as this is the medium through which this community is interacting with its lignocellular-lytic substrate. So the meta-exoproteome is what I'm going to discuss in more detail here. We also extracted nucleic acid, and we were able to use that for community profiling. Thankfully, other people have covered the ins and outs of doing amplicon sequencing. So we did 16 and 18S amplicon sequencing from the DNA. The RNA we used for metatranscriptome library preparation using RNA-seq. And then thirdly, the meta-exoproteome. We looked at this by looking at 2 fractions. We were interested in the soluble proteins, so these are the proteins excreted into the culture supernatant. So we separated the solid fraction, so that's the wheat straw and anything attached to the wheat straw, and we looked at the soluble fraction, so the supernatant, separately. With the solid wheat straw fraction, we used a tagging methodology. So we wanted to look at proteins that were bound to cell surfaces and potentially to the wheat straw surface itself. So we used a biotin labeling technique. Biotin is it's a non-specific protein label, and we used a form of this that is impenetrable to cell walls, to cell membranes. So it will only bind to proteins on cell surfaces or externally to the cell and potentially bound to the wheat straw. Once we'd labeled these proteins, we were able to use a more stringent extraction method, and that lies the total proteome, and then we were able to extract the exoproteomic fraction from that using affinity chromatography. So we ended up with 2 pools of our meta-exoproteome, the soluble and the biotin labeled fraction. And we further analysed the data from week 1, week 3, and week 6, and that's what I'm going to talk about next. So some community diversity stuff. We looked at the phylum level for the bacteria, and it was dominated by proteobacteria and bacteroidetas. And within that, we saw that these were dominated by cytophagia and saprospirae, and we found predominantly alpha and gamma proteobacteria. At the general level, we found that there were distinct abundance groups at week 1, week 3, and week 6. Week 1 was dominated by Astica coli, week 3 Ledbettera, and Tupera in week 6. Astica coli and Ledbettera characterized species within these groups are known to have lignocellular-lytic potential, so that was maybe to be expected, but the Tupera group contain species that are extremophiles, so they're tolerant to high salt, high alkalinity, and that might be reflective of the fact that we saw this increase in pH in our cultures. The liquid cultures were much less diverse than our inoculum, and we can see this in our Shannon index on your right, my left. We did also look at the eukaryotic community, and the inoculum was quite different from our liquid cultures, and our liquid cultures were dominated by this ciliate protist in the alveolata group, and we think that this is due to changing from a solid to a liquid culturing medium. We were interested in which taxa were contributing to the meta-exoproteome that I talked about before. And it seems that mostly these are bacterial proteins. There is a contribution from these alveolata, and I think that's because they're just such a dominant group in the first couple of weeks of the culture. And particularly in the bound fraction and the supernatant fraction, it's dominated by bacterial proteins. We also looked at which taxa were contributing in the bacterial portion. And we found an opposite effect to what we saw at the community level. I remind you, at the community level, we saw mostly Bacteroidetes being the predominant group and the Proteobacteria being the second most dominant group. But it was reversed in the meta-exoproteome. We see more we see more proteins from the Proteobacteria in the meta-exoproteome and a smaller contribution from Bacteroidetes. So to look at, we were interested in functionality of our meta-exoproteomes. So just to go back to this slide and talk a little bit about enzymes that are involved in the degradation of lignocellulose. These are known collectively as K-zymes. They're carbohydrate active enzymes, and there are a huge number of them, and I'm not going to talk in detail. Suffice to say that there's lots of different ones, and we were looking for these kinds of proteins in our meta-exoproteome. We didn't really find them. This was quite surprising to me. We thought they would make up a significant portion of this meta-exoproteome, as we hypothesized that these would be the types of enzymes that would be doing the work of degrading the lignocellulose that I showed earlier. There was mostly glycosyl hydrolases in the bound fraction in week 1, and there's a different pattern that we saw in the soluble supernatant fraction in week 3 and week 6. But overall, they were a very low percentage, less than 3 and 4%. And when we looked at which taxa these proteins came from, quite often they were from taxa that we didn't detect at all in our community profiling work, or that they were at very low percent. So we weren't seeing a kind of correlation between our community abundance data and what we were seeing in our meta-exoproteome. So to briefly summarize, we think we can identify a unique pool of proteins using our tagging approach, and these potentially might be interesting or functional proteins, and this might give a unique perspective on a mixed microbial community. We identified a huge number of proteins, 40% of which have no characterized similarity to any other proteins. And combined with this low number of these Kz enzymes, we suspect this means we don't understand a huge amount about the community degradation of lignocellulose, and that potentially this functional proteome does not correspond well with community abundance measures such as 16S profiling. I would like to thank particularly Dr. Anna Alessi, who led a lot of this work, our collaborators at the University of São Paulo, Igor Polikarpov's group, for doing some of the biomass analysis work, and Neil Bruce and Simon McQueen Mason and Peter Young were the PIs on the project. And I would like to thank everybody for their attention.
Does anyone have any questions?
One thing that jumped out at me there was the massive rise in the pH. I wonder, is there anything about the chemistry related in the degradation of lignocellulose that would lead you to expect that, or if any of the enzymes you saw have very high pH ups in that? I wonder if you could comment.
Generally speaking, it's considered to be unusual that pH rise. A lot of people who we spoke to said that they wouldn't have expected to see that. Certainly fungi that are known to be involved in this process are often acidifiers. So it's one of the question marks that's still hanging over the project, actually. And it does, so if you look at the literature to do with how they industrially process biomass for second-generation biofuels, they actually use alkaline pretreatments to mobilize the lignin and make the other elements more accessible. So I don't know if the two things are connected, but yes, we don't really know why that's happening.
Thanks for a lovely talk. It's so good. I just wondered if you had any thoughts of why you find such a mismatch, and what organisms were producing the enzymes and what organisms did you actually quantify in your communities?
I come at this as quite a naive, I'm not a microbial ecologist, but I suppose it doesn't surprise me very much they're different measures and you're looking at different things, and I think they're both valuable ways of looking at a community. But I think it's quite, it suggests to me that there's maybe an important multiple perspectives that you can get on a community by looking at it from these different perspectives, because each has their own limitation and each has their own technical limitations. You know, there's problems with certain extraction techniques. You maybe won't extract all the DNA uniformly from a community. You maybe won't amplify everything uniformly if you're looking at amplicons. If you're extracting proteins, you don't know what you're extracting and you're relying on, especially if you're identifying things from a very uncharacterized community, you're relying on making your own transcriptome library, which has its own challenges. So my personal feeling is that I think if you want to understand a community, having those different lenses to look at it from could be quite useful and informative. Oh, absolutely. One of the things I didn't have time to present here was actually one of the biggest groups of proteins that we were seeing, which was really surprising actually, was transporters. And lots of different sorts of membrane transporters. And I think there are probably two different groups of organisms. There are ones that are actually actively degrading the lignocellulose, and then there's another group of that are just opportunistically taking up those degradation products.
If you'd like to join me in thanking both Susanna and all the speakers who spoke today for a actually fantastic session. Thank you very much all for coming. Enjoy your lunch. Okay, 30. Good. Now it's 2 weeks. So I'm very curious about how you're assigning causality there. Because you've got data that says that the microbial community of aphids is like this, the community of plants like this, and so how you actually justify saying that there's this directional relationship.
I want to do this with the networks. Only the networks can explain this. Otherwise, just.
I'm really interested in how, that network technique is going to be very interesting what the results are going to come out.
Yeah, now we can see some effects. You saw that.
Yes, yeah. Just it's causality going on.
Yeah. But how actually they interact with each other, I can see only with networks, I think. So that will be the next step.
It'll be absolutely fascinating to see those results.
Yeah, I get the results a few months ago. Just the last chapter of my thesis, so I get the results there and write the other part of my thesis, and then I finished to write this like 3 weeks ago. So these are sort of preliminary results.
How far have you finished that?
I delivered the thesis like 4 days ago.
Congratulations.
Thank you. And so my defense will be in February, more or less. But we'll start my postdoc in January. Yeah.
the